Put 2 fingers on the side of your neck. Feel your pulse. Don't count it. Don't analyze it. Just notice how fast it's moving right now, while you're sitting here, watching this. That rhythm under your skin is the gatekeeper to sleep. If it stays elevated, you cannot become unconscious. Not truly. Not in the way your brain requires. No matter how many hours the clock accumulates on the other side of the room. 8 hours won't save you if that pulse never slows. Sleep is not a number. Sleep is a threshold. And almost everything you have been told about how to cross it is wrong. But the deeper problem, the one nobody addresses, is not just how you fall asleep. It is what happens after. Because you can cross that threshold and still fail completely. You can be unconscious for 8 hours and wake up with your brain in worse condition than when you closed your eyes. The hours are not the program, the sequence is. And almost no one has told you what the sequence actually does. Here is where the lie begins. In the 1930s, researchers observed healthy young adults sleeping in controlled conditions. They measured how long these people naturally slept when left undisturbed. The average came out to approximately 8 hours and 13 minutes. That number entered the scientific literature. And somewhere between that laboratory and the morning talk shows and the wellness blogs and the doctor's offices, the variance disappeared. The range became a rule. The average became a commandment. 8 hours. But here is what that study actually showed. Some people needed 6.5. Some needed 9.25. The 8 hours was the middle of a distribution, a description of a crowd, not a prescription for an individual. And more critically, it said nothing, absolutely nothing, about what was happening inside those bodies during those hours. Because you can spend 8 hours in bed and never once cross into the sleep your brain needs to function, recover, and survive. The question was never how long. The question was always whether. Whether the right stages ran. Whether they ran in order. Whether anything interrupted them before the work was done. Stop counting the hours. Start understanding what happens inside them. Think about this morning. Not the alarm. The moment before it. Or the first few seconds after. Before you moved. Before the day began assembling itself in your mind. There was a feeling. Heavy, slightly underwater, the room present, but you not yet fully inside it. Thoughts arriving slowly, as if from a distance. You have probably spent your entire life interpreting that feeling as evidence of insufficiency. Not enough hours. Need more. The number was too small. It is not insufficiency. It is the evidence of completion. What you were feeling in those first seconds was the final phase of a biological system switching off. A system that spent the night doing something your brain cannot do while you are awake, in any waking state, under any circumstance. When that grogginess clears in 15 to 20 minutes and your thoughts arrive at normal resolution, that is what a finished program feels like from the inside. When it persists for hours. When it is replaced by a flatness that no amount of coffee reaches. When you feel more tired after 8 hours than you did before them, the program fragmented. The hours were counted. The function was not delivered. To understand why, you need to understand what the program actually is. Every night, if nothing breaks it, your brain runs 4 to 6 cycles. Each cycle lasts approximately 90 minutes. Each one contains a specific sequence of stages, and each stage performs work that no other stage can replicate, work that cannot be recovered once missed, cannot be rescheduled, cannot be made up on the weekend. The sequence is not negotiable. Break the order, and the hours remain while the function disappears. Here is what runs inside each cycle. Here is what you are actually losing when the architecture collapses. The first stage is the descent. None one. The lightest sleep. The transition between wakefulness and unconsciousness. Your heart rate begins to fall. Your muscles release their held tension. Your eyes move slowly beneath their lids. Hypnic jerks, that sudden falling sensation that snaps you awake, occur here, a quirk of a nervous system learning to let go. This stage lasts only minutes. It is the doorway, not the room. But the doorway only opens if one thing has happened first. Your core body temperature must fall. Not dramatically. Not uncomfortably. Approximately half a degree to a full degree Celsius. Your body achieves this by opening the blood vessels in your hands and feet. Heat moves from your core to your skin and radiates outward. This is why your hands and feet feel warm just before you fall asleep. It is not comfort. It is your body opening its radiator valves, moving heat from the inside out. As your core temperature falls, your pineal gland, a pea-sized structure buried in the center of your brain, begins releasing melatonin. Melatonin is not a sedative. This is one of the most consequential misunderstandings in popular sleep science. Melatonin does not drag you into unconsciousness. It is the ignition key. It signals every cell in your body that biological night has arrived and that the conditions for sleep are being met. The key turns, the engine starts. But if your core temperature doesn't fall, because you ate a heavy meal too late, because you drank alcohol, because your room is too warm, because you spent the last hour under bright lights, your melatonin production stalls. The key doesn't turn. The engine doesn't start. You lie in bed for 8 hours. The clock runs. The biology doesn't. 8 hours is a statistic. The temperature drop is the event. The second stage is where sleep becomes real. NEN2. Your brain begins producing sleep spindles, rapid bursts of neural activity, oscillating at 12 to 15 hertz, generated by your thalamic reticular nucleus. These spindles are not random. They are the brain's deliberate act of muting incoming sensory information, building a wall between you and the world, so the deeper work can begin undisturbed. Your heart rate drops further. Your breathing slows. Your body temperature continues its descent. And your brain begins its first pass at memory. Everything you learned today, every conversation you had, every skill you practiced, it entered your hippocampus as a raw, fragile trace, temporary, vulnerable. In N2, the first sorting begins. The hippocampus starts flagging what matters. The spindles create the quiet it needs to work. This stage makes up nearly half of your total sleep time across the night. It is not glamorous. It does not appear in the headlines. But it is the stage most consistently stolen by alcohol, by late caffeine, by anxiety, that keeps the thalamus from building its wall properly. When N2 is fragmented, you lose the quiet. And without the quiet, the deeper stages cannot load. The third stage is the maintenance window. N3. Slow wave sleep. The deepest stage your brain enters. And the one that almost everything you have been told about sleep has failed to mention. During slow wave sleep, something happens inside your brain that neuroscience did not have the tools to see until 2013. Maiken Nedergaard was a neuroscientist at the University of Rochester. She had been studying astrocytes, the star-shaped glial cells surrounding neurons, for years. Astrocytes were considered scaffolding, the cells that held neurons in place, supplied them glucose, maintained the architecture, structural, not interesting. Nedergaard was not convinced. Her team injected fluorescent tracers into the cerebrospinal fluid of sleeping mice and watched what happened under a 2-photon microscope, a device capable of imaging living tissue at the cellular level in real time. The tracers moved through brain tissue rapidly, in a directed pattern, flowing in through channels surrounding arteries, sweeping through the spaces between neurons, draining out through channels surrounding veins into the cervical lymphatics. This should not have been possible. The brain has no lymphatic system. That was not a gap in the literature, it was established anatomy. The absence of a lymphatic system in the brain had been confirmed and accepted for a century. Her team spent months ruling out artifacts. Equipment malfunction, methodological error. The movement was real. The flow was real. Here is the mechanism. During slow-wave sleep, your astrocytes actively shrink, not passively, actively, driven by ion transport, a deliberate cellular contraction. They shrink by approximately 60% of their resting volume. 60%. The interstitial space between your neurons expands from approximately 14% of total brain volume to approximately 23%. Your brain physically opens, and into those expanded spaces rushes cerebrospinal fluid, sweeping through the tissue, carrying something out: beta amyloid, tau protein, lactate, the metabolic exhaust of every thought, every decision, every moment of attention you sustain today. Your brain is washing itself. Glymphatic clearance during slow-wave sleep is 10 to 60 times more active than during waking hours. Not incrementally more, 10 to 60 times. Nedergaard named it the glymphatic system, published in Science in 2013, replicated across multiple independent research groups since. Sleep was not rest. Sleep was the nightly sanitation of your brain. And in 2018, researchers at the National Institutes of Health used PET scanning to measure beta amyloid burden in human brains before and after a single night without sleep. Not a week of deprivation, not a month, one night. The accumulation was measurable, visible on the scan, concentrated in the hippocampus and thalamus. the structures most critical for memory and consciousness. One night, your brain produces this waste during every waking hour of cognition. The glymphatic system clears it during slow-wave sleep. Miss the maintenance window through alcohol, through stress, through a room that is too warm, through a schedule that never lets the architecture run deep enough, and the clearance falls behind the production. The deficit grows slowly, below the threshold of morning symptoms, below the resolution of any sleep tracker, in the way that structural damage accumulates, quietly, persistently, with nothing announcing itself until the threshold has already been crossed. Here is where the stakes become impossible to ignore. Beta amyloid is the primary protein constituent of the plaques found in the brains of people with Alzheimer's disease. In January 2023, the FDA approved lecanemab, brand name Leqembi, for early Alzheimer's disease. To receive it, 4 infusion every 2 weeks in a clinical setting. A prescribing neurologist. Baseline MRI, repeat MRI at weeks 26 and 52, approximately $26,500 per year, before insurance. Risk of brain edema in 12.6% of patients, microhemorrhages in 17.3%. In early-stage patients, a 27% slowing of clinical decline, not reversal, slowing. The most expensive new drug in neurology is attempting to do chemically, at the end of a decades-long accumulation process, what your brain's slow-wave maintenance window was designed to prevent from the beginning. $26,500 a year. The alternative ran every night, free, until the architecture broke. The 4th stage changes what sleep means entirely. RAM. RAM. Some rapid eye movement sleep. The stage most people have heard of. The stage almost no one understands correctly. In your brainstem, there is a structure called the locus coeruleus. It contains approximately 1,500 neurons per side, among the smallest named nuclei in the brain. It is the primary source of norepinephrine, the neurochemical of stress response, threat detection, sustained alertness for the entire brain. During REM sleep, the locus coeruleus goes completely silent. This is the only sleep stage in which this happens. Norepinephrine drops to 0. And in that chemical silence, your amygdala, highly active during REM, replays the emotionally charged experiences of your day in a brain temporarily freed from stress chemistry. The charge of difficult memories is processed, reduced, integrated. The brain represents the day's hardest material in the one neurochemical environment where it can be handled without the stress response firing. This is why you wake from interrupted REM with emotions still present in your body, still raw, still unresolved. The processing was incomplete. The memory was replayed but not resolved. The wound was reopened without being treated. 1,500 neurons per side. That is the structure whose silence enables your brain to process grief, fear, frustration, and loss overnight. 1,500 neurons going quiet so the work can happen. REM does something else that slow-wave sleep cannot. While slow-wave sleep consolidates individual memories, REM integrates them. It finds the hidden connections between things you learned today and things you learned months ago. It builds the map. This is why problems you sleep on have solutions in the morning that were invisible the night before. This is why creative insight follows a good night's sleep with a consistency that is not metaphorical, it is neurological. REM is running pattern recognition across your entire memory architecture while you dream. And REM is not distributed evenly across the night. REM is concentrated in cycles 4, 5, and 6, the final 90 minutes to 3 hours before your natural waking time. Cut the last 90 minutes. Set the alarm 90 minutes earlier than your body would naturally rise. You do not lose a proportional slice of every stage. You lose almost exclusively REM, the emotional processing stage, the pattern recognition stage, the memory integration stage, the stage that 1,500 neurons per side spend the whole night preparing for. 6 hours ending at the right biological moment can deliver more REM than 8 hours cut short by an alarm. The number is not the program. Now consider what breaks the program. Specifically, predictably, in ways the number 8 will never detect. Alcohol, not a sleep aid, a program destroyer with excellent marketing. Alcohol increases GABA receptor activity in your brain. It quiets neural firing, the world softens, your eyelids drop, you fall asleep faster. This part is real. But sedation is not sleep. Alcohol suppresses REM in the second half of the night by up to 25%. And as your liver metabolizes the alcohol across the night, the byproduct, acetaldehyde, is neurologically stimulating. It fragments your slow-wave architecture from the inside while you believe you are sleeping through it undisturbed. 2 glasses of wine at 10 p.m. means active metabolizing until midnight, acetaldehyde disruption from midnight to 2 a.m., fragmented slow-wave sleep from 2 a.m. onward, and REM suppressed through the final cycles entirely. Your sleep tracker sees 8 hours. Your locus cerebellosus never got quiet long enough to do its work. Your glymphatic system never fully opened. Your hippocampus never finished the transfer. You think you slept 8 hours. Your brain experienced something closer to 4. Caffeine afternoon. Caffeine occupies adenosine receptors, blocking the signal of accumulated cognitive fatigue. Its half-life is 5 to 6 hours in average adults. Longer, up to 9 hours in some individuals. A coffee at noon carries meaningful receptor occupancy at 10 p.m. Your sleep pressure signal is partially suppressed. The first slow-wave cycle, the opening of the glymphatic maintenance window, is shallower. The brain's cleaning system does not fully open. The hours of sleep will be there, the depth will not. Chronic elevated stress. Sleep deepens through a thermoregulatory mechanism. Your core body temperature must fall to trigger the transition into slow-wave sleep. Sustained psychological stress elevates baseline cortisol. Elevated cortisol prevents the temperature drop. The thermoregulatory trigger for the maintenance window is blocked. The slow-wave threshold is never reached. The sleep tracker records 8 hours of sleep. The architecture records 8 hours of stages that never reach the floor. Sleep apnea. Microaerosols of 3 to 15 seconds. Dozens to hundreds of times per night, of which the person sleeping is completely unaware. The glymphatic window opens and closes before it can clear. The locus cerebellosus cannot sustain its silence. The person wakes after 8 hours certain they slept. Their brain ran an interrupted program for 8 hours, and completed almost none of it. Your brain does not count hours. It executes a program. The program either finishes or it doesn't. Now let's talk about the 3 a.m. problem, because it sits at the exact center of everything you've just learned. Almost everyone watching this has experienced it. You wake up suddenly. The room is dark. You feel a strange, uncomfortable alertness. Not rested, not sleepy, just awake. You look at the clock, 2:17, or 3:08, or 3:44. Your first thought, something is wrong with you. Your second thought, it's aging. Both are wrong. This waking is built into your biology. Your core temperature has been falling since you went to sleep. It reaches its lowest point around 1 to 3 a.m. At that point, your body triggers a slight thermal rebound, a small deliberate rise in core temperature to protect you. That rebound produces a brief surfacing from sleep. For most people, this lasts 30 seconds. Heart rate stays low, they roll over, they disappear back under, they never remember it. For some people, something goes wrong in those 30 seconds. They reach for their phone. The light from that screen activates melanopsin-containing photoreceptors in your retina. Those cells send a direct signal to your suprachiasmatic nucleus, 20,000 neurons in your hypothalamus, functioning as your master circadian clock. Your clock interprets that light as dawn. Melatonin production stops immediately. And then you do something even more destructive than the light. You do the math. It's 3:30. I have to be up at 6. That's only 2.5 hours. If I don't fall asleep right now, tomorrow is completely ruined. That thought activates your amygdala. Your amygdala signals your adrenal glands. Cortisol and adrenaline enter your bloodstream. Your heart rate climbs. The thermal rebound accelerates instead of resolving. The glymphatic window that was briefly interrupted cannot reopen against that cortisol tide. You have just locked yourself out of the remaining REM cycles. The final stage. The one your brain spent the entire first half of the night preparing for. The 3 a.m. waking is not your problem. The phone is the problem. The math is the problem. Here is what you do instead. Do not reach for your phone. Do not look at the clock. Time is irrelevant at 3 a.m. Only threshold is relevant. If you feel the urge to use the bathroom, do not immediately stand up. Roll onto your back first. Wait 25 seconds. The urge will often dissolve. Body weight on the bladder when side sleeping creates false urgency signals. Standing raises your heart rate immediately, and returning it to threshold takes 10 to 15 minutes. If you must go, do not turn on the overhead light. A small night light at floor level, placed before bed, prevents your suprachiasmatic nucleus from receiving the dawn signal that stops melatonin cold. Then use the 4-7-8 breath. Breathe in through your nose for a count of 4. Hold for 7. Breathe out through your mouth for 8. The long exhale is the mechanism. It forces your vagus nerve to fire, which tells your sinoatrial node to slow down. Your heart rate drops back toward the threshold. Not because you forced it, because you gave your nervous system the physical signal it needed to release. Do 20 cycles. Track them on your fingers. Visualize each number as you count. See the 4, see the 7, see the 8. Here is why this works beyond the breathing. You cannot count and catastrophize at the same time. The anxious math and the breath count compete for the same neural resources. You cannot run both processes simultaneously. The count wins. The cortisol response dissolves. The thermal rebound completes naturally. The remaining REM cycles become available again. You are not forcing sleep. You are removing the obstacles standing between you and it. So what does all of this mean for tonight? Not a list. A reframe of the question itself. The question your sleep tracker asks every morning is, how many hours was this person unconscious? It is a tractable question. It is not the right question. The right question is whether the program completed. 3 variables address that question directly. Protect the end of your night. REM lives in the final cycles. An alarm set 90 minutes earlier than your body would naturally wake cuts disproportionately into the stage responsible for emotional processing, pattern recognition, and memory integration. When your schedule allows, protecting the end of the night recovers more function per minute than adding equivalent time at the beginning. Understand that tonight's architecture is being written this afternoon. The caffeine you consume after noon suppresses the adenosine pressure that determines how deep your first slow-wave cycle goes. The meal you eat 2 hours before bed generates the heat that stalls melatonin production. The alcohol you drink at 10 p.m. will fragment your REM at 3 a.m. while you sleep through the damage completely. These are not restrictions, they are mechanisms. The architecture of the program running tonight is being determined right now, hours before you close your eyes. When you wake at 3 a.m., remember what it is. Your body is running its intersleep interval, the thermal rebound completing. The transition between first sleep and second sleep that humans ran for centuries before artificial light compressed the night into a single block and handed the waking to you as a medical disorder. The calm of those first 30 seconds before the calculation begins, that is your biology working correctly. The panic that follows is the only thing breaking it. Put 2 fingers back on the side of your neck. Feel your pulse. Is it slower than when you started? It might be. You have been sitting still. You have been breathing without urgency. That slight deceleration under your fingers is your vagus nerve doing its work. The warmth in your hands, if you notice it, is peripheral vasodilation, your body already moving heat from core to surface. Already beginning. Your biology is not broken. It has not failed you. It has not declined into something unfixable. It has been waiting, precisely, patiently, without complaint for the conditions you now know how to create. The 8-hour rule was never about your biology. It was about a crowd of people whose average was mistaken for a universal law. You are not an average. You are a specific sequence of stages, thermal, cardiovascular, neurochemical, that when run in the right order and allowed to complete produce something no number on a clock has ever been able to measure. A brain that washed itself overnight. Memories that transferred. Emotions that processed. The architecture ran. The program finished. Stop counting the hours. Start protecting the sequence. The grogginess you feel some mornings, that heavy, slightly underwater feeling before the day assembles itself, is the glymphatic system completing its final clearance cycle. The maintenance window closing as consciousness returns. Not evidence that you needed more hours, evidence that something happened during the ones you had. The vivid dream your alarm cut into was 1,500 neurons per side gone completely silent, your amygdala processing the emotional residue of your life in the only neurochemical environment where it can do so without the stress response firing. And the alarm arrived before the sentence finished. And the morning when your body was returned to you cleanly, before the alarm, eyes opening into a room already in focus, thoughts arriving at normal resolution, the day beginning without resistance, the program ran. Every stage, in order, to completion. Not because of the number, because of the sequence. The program is running tonight. The only question is whether it finishes.